Somewhere inside a muscle fiber, a small molecule crosses a membrane, and everything downstream of that single event starts to shift. Creatine doesn't diffuse impassively. It needs a specific carrier called the creatine transporter, known in the scientific literature as creatine, to pull it across the cell membrane and into the muscle fiber. This transporter is sodium dependent and can become saturated, which is one of the reasons why simply consuming more creatine doesn't translate into unlimited accumulation inside the cell. Once inside, the vast majority of that creatine doesn't stay in its free form for long. Roughly 2/3 of it gets phosphorylated and stored as phosphocreatine, a high-energy reserve tucked inside the cytoplasm of the muscle fiber, ready to be called upon the instant the cell needs a rapid burst of energy. The remaining portion stays as free creatine, acting almost like a buffer that keeps the phosphocreatine system running smoothly during exchanges of energy. What's striking is how old this molecule's story actually is. Creatine was first identified back in 1832 by the French chemist Michel-Eugène Chevreul, who found it in meat extract and named it after the Greek word for flesh. Almost 2 centuries later, researchers are still mapping exactly how it behaves inside a living human muscle cell. And the first 24 hours after supplementation begins are where that behavior starts to become measurable. Before any visible change in muscle size shows up, something else happens first, and it's often misunderstood. Creatine is osmotically active, meaning it draws water toward itself. As creatine concentration rises inside the muscle cell, water follows it across the membrane from the surrounding fluid, and the cell physically swells. This is not the same as the kind of water retention people associate with feeling puffy or bloated under the skin. This swelling happens specifically inside the muscle fiber itself. Researchers Dieter Heusinger, Enrico Roth, Florian Lang, and Wolfgang Gerock published a paper in The Lancet in 1993, describing something that reframed how scientists thought about hydration at the cellular level. Cell volume itself functions as a signal. When a cell swells, it tends to shift toward building processes, increasing protein synthesis and slowing the breakdown of existing proteins. When a cell shrinks, the opposite tends to happen, tilting the balance toward breakdown. Under that model, the swelling triggered by creatine isn't just cosmetic. It's information the cell is receiving about its own internal state. This is part of why some people notice the number on the scale creep up within the first few days of supplementation, sometimes by 1 to a few pounds. That shift reflects water being pulled into muscle tissue, not fat accumulation. It also explains why muscles can look and feel fuller within days, well before any structural change like new muscle protein has had time to accumulate. To understand why creatine matters so much to a working muscle, it helps to understand what a muscle cell actually runs on. Every contraction depends on ATP, the molecule that supplies usable energy, but a cell's available ATP is tiny, so small that if nothing replenished it, contractions would grind to a halt within 1 to 2 seconds. This is where phosphocreatine becomes essential. It donates a phosphate group to spent ADP molecules, regenerating them back into ATP almost instantly. During intense muscular effort, phosphocreatine levels inside the muscle can fall by close to 50% within just 6 seconds, which shows how quickly this reserve gets called upon during short, powerful bursts of activity like a heavy lift or a sprint. Philip Greenhalgh and colleagues, publishing in the American Journal of Physiology in 1994, demonstrated that supplementing with creatine doesn't just raise the total amount of creatine in the muscle, it also speeds up how quickly phosphocreatine gets resynthesized after it's been used. That faster resynthesis matters most in activities defined by repeated short bursts rather than sustained endurance output. Think of repeated heavy sets in a workout rather than a long, steady run. Because phosphocreatine can be replenished more rapidly, the muscle has more of that instant energy currency available for the next contraction, which is a large part of why creatine's effects show up first as a change in performance capacity rather than a change in appearance. Muscle tissue doesn't absorb creatine indefinitely. There's a ceiling, and research has mapped out roughly how long it takes to approach it. Eric Holtman and colleagues, publishing in the Journal of Applied Physiology in 1996, tracked 31 men taking creatine at different doses and found that muscle total creatine concentration rose by approximately 20% after 6 days when intake was around 20 grams per day. That elevated level held steady when intake was then reduced to about 2 grams per day for a further 30 days. The same study found something equally informative. Taking a smaller amount, around 3 grams per day, produced a similar 20% rise in muscle creatine. It just took about 28 days rather than 6 to get there. In other words, the muscle arrives at roughly the same saturation point either way. What differs is the speed of the climb, not the final destination. This is one of the clearer, better replicated findings in the creatine literature, and it forms the backbone of why day 7 tends to mark a turning point in most supplementation timelines that use a faster loading approach. Not everyone starts from the same baseline either. People with lower resting muscle creatine levels before supplementation, a group that includes many vegetarians, since dietary creatine comes primarily from meat and fish, tend to show a larger relative increase once supplementation begins. This individual variability is one reason researchers are cautious about describing a single universal timeline, even though the general saturation window around 1 week for higher intakes has been observed consistently across multiple studies. We already saw that creatine pulls water into the muscle cell, causing it to swell from the inside. What matters now is what that swelling actually does once it happens. Because a cell's hydration state isn't just a passive container effect, it functions as information the cell reads and reacts to. In the framework proposed by Heusinger and colleagues, an expanded, well-hydrated cell tends to shift its internal chemistry toward building processes, including the activation of glycogen synthase, the enzyme responsible for storing glucose as glycogen inside the muscle. At the same time, that same hydration state appears to suppress the pathways responsible for breaking proteins down. So the net effect of a swollen muscle fiber isn't simply more water. It's a cell that has shifted its internal balance slightly away from breakdown and slightly toward maintenance and construction. Researchers describe this as an anabolic bias, a subtle tilt in the scale rather than a dramatic, immediate transformation. And it's a mechanism that operates independently of whether a person is exercising or completely at rest. This is one reason creatine's effects show up even in people who aren't training intensely, though the effect compounds substantially when resistance exercise is added into the picture. The mechanical tension and metabolic stress created by lifting weights appears to work alongside this hydration-driven signal rather than replacing it, which is part of why the combination of creatine and structured training produces different outcomes than either factor does alone. Before we keep going, let's pause for a second. If you already take creatine or you've thought about trying it, tell me down in the comments. Did you notice that fuller, pumped feeling in your muscles during the first few days? Write down what you felt. Muscle tissue isn't uniform. It's made of different fiber types with distinct jobs, and creatine doesn't affect all of them equally. Type 2 fibers. The fast-twitch fibers responsible for short, powerful, explosive contractions naturally carry a higher concentration of creatine phosphokinase activity and rely more heavily on the phosphocreatine energy system than type I, slow-twitch fibers, which are built for sustained, lower-intensity work and depend more on oxidative metabolism. This difference in fiber biology helps explain a finding that surprised researchers studying individual variation in response to supplementation. Daniel Syratuik and Gordon Bell, publishing in the Journal of Strength and Conditioning Research in 2004, examined 11 men undergoing a 5-day creatine loading protocol and found that those who responded most strongly, with the largest increases in muscle creatine content, had an average of 63.1% type 2 fibers in their vastus lateralis muscle. Those who barely responded had an average of just 39.5%. That gap in fiber composition offers a partial explanation for something almost everyone taking creatine eventually notices. The experience isn't identical from person to person. Someone with a naturally higher proportion of fast-twitch fibers may notice changes in fullness and power output more quickly within the first 2 weeks, while someone with a fiber profile weighted more towards slow-twitch tissue may experience a more gradual, less dramatic shift over the same period, not because anything is wrong, but because the underlying muscle architecture is different. One of the more counterintuitive parts of this story is timing. Visible muscle growth requires weeks of accumulated training stimulus, but changes in performance capacity can show up much sooner, sometimes within the first several days. This isn't a coincidence. It follows directly from the phosphocreatine mechanism covered earlier. If the muscle has more readily available energy for short bursts of maximal effort, that advantage in output can appear before there's been enough time for meaningfully more muscle protein to be built. Philip Greenhalgh and colleagues demonstrated this back in 1993, showing that after just 5 days of oral creatine supplementation, subjects performing repeated bouts of maximal isokinetic contractions maintained torque output better across those repeated efforts compared to a placebo group. That protective effect was most pronounced in the final repetitions of the first bout and carried through the later bouts of the protocol, suggesting creatine's early advantage shows up less as raw new force and more as resistance to the decline in output that normally accompanies repeated maximal effort. What this timeline reveals is that the phosphocreatine energy pathway responds on the scale of days, while the structural machinery of muscle growth, new contractile proteins, expanded fiber cross-sectional area, operates on the scale of weeks and requires an ongoing training stimulus layered on top of supplementation. Someone tracking their experience over the first 30 days is essentially watching 2 different processes unfold on 2 different clocks. With the faster one, performance, generally becoming noticeable well before the slower one, size. Underneath all the faster moving changes, the water shifts, the energy system refills, the early performance gains, there's a slower process quietly getting underway, one that won't show meaningful results within a single month. Satellite cells are specialized cells that sit on the outside of muscle fibers, largely dormant until triggered by mechanical stress or damage from training. Once activated, some of these cells fuse into existing muscle fibers, donating their nucleus to help support additional cytoplasmic volume, a concept researchers refer to as the myonuclear domain, since each nucleus can only support so much surrounding muscle tissue before more nuclei are needed for further growth. Steen Olsen and colleagues, publishing in the Journal of Physiology in 2006, tracked 32 men through 16 weeks of heavy resistance training and found that the group supplementing with creatine showed a significantly greater increase in both satellite cell number and myonuclei count compared to groups receiving protein or a placebo. This tells us the process is real and measurable. But the timeline the researchers used to detect it was 16 weeks, not 30 days, which matters enormously for how this information should be understood. That distinction is worth sitting with. The first 30 days of creatine supplementation likely represent the earliest phase of satellite cell activation rather than the phase where its results become visible. It's less like flipping a switch and more like planting something that needs several additional months of consistent training stimulus before the structural payoff. Genuinely new muscle tissue, not water, not energy substrate, but added contractile protein, becomes something a researcher could actually measure on a biopsy slide. By the time day 30 arrives, the muscle has been living with elevated creatine levels for a while, though exactly how long depends on which path was taken to get there. Someone following a faster loading approach reached saturation around day 6 or 7, meaning that by day 30, the muscle has already spent more than 3 weeks in that elevated state. Someone following the slower, lower dose approach is likely just arriving at that same roughly 20% increase in muscle creatine content that Holtman and colleagues measured back in 1996, since their data showed the gradual protocol reaching a comparable plateau closer to the 28-day mark. Adaptation at this stage isn't limited to creatine concentration itself. Adil Safdar and colleagues tracked 12 young, healthy men through 10 days of creatine supplementation, using 20 grams per day for the first 3 days, followed by 5 grams per day for the remaining week, without any exercise intervention at all. They found significant increases in both total body water and fat-free mass, and, notably, differential gene expression patterns that the researchers interpreted as a homeostatic adaptation to the change in cellular osmolarity. In other words, the cell isn't just holding more water. Its internal gene activity appears to be responding to that new hydration state. Body composition data collected around the 30-day mark in trained populations tells a consistent story. A study following 18 male powerlifters through 26 days of supplementation, with measurements taken on day 1 and day 28, found significant increases in both body mass and lean body mass, with no corresponding change in body fat percentage. Taken together, this suggests that by day 30, a real and measurable shift in body composition is already present, even though, as covered earlier, the deeper structural process involving satellite cells and new contractile tissue is still only in its opening phase. With 30 days of research now laid out, it's worth directly separating what the data supports from what popular belief has attached to this molecule over the decades. One persistent myth treats creatine as functionally similar to an anabolic steroid, but the mechanisms described throughout this video, a transporter-mediated entry into the cell, a phosphate-donating energy system, an osmotic pull on water, a signal tied to cell hydration state, operate through entirely different biological pathways than synthetic hormones that bind androgen receptors. The overlap in cultural perception doesn't reflect an overlap in actual physiology. A second area of concern people raise involves kidney health, largely because creatine supplementation elevates blood creatinine, a marker doctors use to screen kidney function. The International Society of Sports Nutrition, in its 2017 position, stand authored by Richard Kreider and colleagues, reviewed the accumulated evidence and concluded that both short-term and long-term creatine use, at doses reaching up to 30 grams daily for as long as 5 years, was safe and well tolerated in healthy individuals. The position stand specifically notes that the rise in creatinine reflects normal creatine metabolism rather than kidney damage, a distinction that matters for anyone trying to interpret their own lab results. So what does 30 days of creatine supplementation actually change inside human muscle, according to the evidence gathered across this video? A transporter pulls creatine into the fiber, where it's mostly stored as phosphocreatine. Water follows the creatine inward, swelling the cell in a way tied to hydration-based cellular signaling. The replenished phosphocreatine system allows faster energy regeneration during short, powerful efforts, which is why performance shifts tend to appear before any visible size change. Fiber type composition shapes how strongly any individual responds. And underneath all of it, satellite cells have only just begun the much longer process that, months down the line, produces the kind of new muscle tissue that a biopsy could actually confirm. If this changed how you think about what's actually happening inside a muscle during those first 30 days, Hit like so more people can find this. Subscribe if you want the rest of this series. We're going organ by organ, system by system, always from the research, never from the hype. And if there's someone in your life who takes creatine but has never really understood why it works, share this with them. See you in the next one.